Assalamu alaikum, welcome back to BioTalk with Hajra. Today we will explore DNA replication. How does one cell become two with the same genetic information? Let's begin. What is DNA replication? It is how DNA makes a copy. One molecule becomes two. This occurs before division. It makes an identical copy. One molecule becomes two. This happens before division. The cell copies its DNA. One DNA molecule becomes two. This happens before cell division. A cell must copy its DNA. Each cell gets a copy. A cell cannot simply divide its DNA. It first needs to copy its entire DNA. Then, each cell receives a complete copy. Why is DNA replication important? It's necessary for growth and development. Your body replaces millions of cells. Let's find out. When does DNA replication occur? In eukaryotic cells, it occurs during S phase. Interphase is when the cell grows. S phase stands for synthesis. Today we will study DNA replication. The two strands separate. Each acts as a template. Today we will learn about DNA replication. It is semi-conservative. This means that each strand acts as a template and we get two DNA molecules. Each one contains one old strand, one new strand. The DNA replication begins at a specific region. An enzyme called DNA helicase plays a major role here. Think of DNA like a zipper. This creates the replication fork. Tension builds up ahead of the replication fork. As helicase unwinds the DNA, topoisomerases help relieve that. We are studying DNA replication. Topoisomerase relieves strain. In bacteria, an important strands tend to come back together, so they need to be stabilized. This is done by SSB proteins. They prevent Now we have another problem. DNA polymerase cannot start building a new strand from nothing. It needs a starting point. This is provided by an RNA primer. The primer provides a free 3'OH group. This is what DNA polymerase needs. Now comes DNA polymerase. It adds nucleotides to the strand. It synthesizes only in the 5' to 3' direction. To 3' direction, today we will analyze DNA replication. Pay attention to the 5' and 3' ends. This directionality is extremely important for understanding the leading and lagging strands. Different organisms use different mechanisms for primer removal. In bacteria, DNA polymerase I removes RNA primers. In eukaryotes, enzymes like RNase H and FEN1 are involved. RNA primers cannot remain in the DNA. They must be removed and replaced. Bacteria use DNA polymerase I. Eukaryotes use RNase. Now we have another problem. The lagging strand contains multiple Okazaki fragments. These fragments need to be joined together. It joins the pieces into one continuous strand. This is where DNA ligase comes in. DNA ligase seals the breaks between DNA fragments by forming phosphate. Think of DNase, it joins the separate pieces into one continuous DNA strand. DNA replication must be extremely accurate. Fortunately, DNA polymerases have proofreading mechanisms. They detect and remove incorrect nucleotides. This ability increases accuracy. What happens at the end of replication? The result is two DNA molecules, one original and one newly synthesized strand. That is why it's called semi-conservative. Telomeres are at linear chromosome ends. Normal machinery cannot fully replicate this very end. This is called the end replication problem. Telomerase helps maintain these sequences. Telomeres are at linear chromosome ends. Normal machinery cannot fully replicate this very end. This is called the end replication problem. Telomerase helps maintain these sequences. DNA replication is a highly coordinated process. It involves multiple enzymes like helicase and polymerase. One molecule becomes two. That is the incredible science of DNA replication. If you found this helpful, like, share and subscribe to BioTalk with Hajra. Join us for simple biology and microbiology. Thank you for watching and see you next time.